So we are going to have a quick review about the enzymes. As we know that enzymes are biological catalysts and they speed up the reactions. So they support chemical reactions. Living cells will depend upon these cells, which are enzymes, and by nature, chemically they are proteins. We will talk about the action and nature of enzymes. So they lower the activation energy. The reactions which are being taken place by the enzymes are reversible and they speed up the reaction in both the directions. So we will have enzyme that reacts with the substrate and it gives the products. So that is what we are going to focus on. So they are temperature and pH specific. In addition to that, they form an enzyme and substrate complex and they try to speed up the chemical reaction without getting utilized, which means the enzymes can be recycled. So, enzymes are recycled and they will be free to combine with the new amount of substrate and converts it to the products. Now we will try to understand how the structure of enzyme is. So as we know that enzymes are the proteins, so we are aware that proteins are nothing but the polypeptide chain, which are polymer of amino acids. So they are formed by the amino acids and they fold and they form such structures which we can consider as a polypeptide chain. So because of the folding of polypeptide chain, it forms an active site and that active site specifically binds to the substrate and that results into enzyme substrate complex and then it forms enzyme and the product will be formed. So this is how the reaction happens. For that, we can understand the lock and key model, like the every key is specific for the lock. Similarly, the enzyme is specific for the substrate by because of its active site. So its active site has specific groups which can combine with this substrate and that substrate combines with the enzyme to form enzyme substrate complex and then it can get converted into the product. So that is lock and key model. While so this is the hypothesis which explains how enzymes combine to the substrate and they convert into the enzyme substrate complex and they form products and the enzyme is recycled. So enzyme is now free to recombine with the new amount of substrate. The mechanism behind the enzyme's activity is you can resemble a situation like you have gone on a walk, you have to cross this hill. So what do you require? You require that the amount of energy so that you can climb this hill and then only you can get down and you may reach to the other side of the hill. The same thing applies here. So here when the substrate is getting converted into the product, it must have the amount of energy which can convert this to the product. So this is the amount of energy of the enzyme. So this requires this much energy in order to get converted into the product. But when you add the enzyme to this, now this energy is minimized or this energy has been lowered down. And now what can be done with the help of that? The amount of activation energy has been lowered down. So the reaction becomes faster and quicker, right? So that is why our product will be formed very easily. So with the help of this, we can understand the same thing like when there are two graphs have been shown over here. So that's the energy of the substrate, energy of the product is somewhere here. And this is the transition state where your enzyme substrate complex would be formed. So here there is no enzyme, just the substrate has to overcome this. And this much is the activation energy with enzyme. So when you add an enzyme, the activation energy has been lowered down. And as a result, your product formation is favored. So that's what it happens when you add an enzyme. Now we have talked about lock and key model. Now this one is induced fit model, which says that yes, active site is there, which combines with the substrates, but in addition to that, there is one more aspect that the active site can make a little conformational changes. It can change its shape so that it binds to the substrate. So even if it is not specific for that, has a little change in the shape, but it can adjust its shape so that it can fit into this and can convert the substrates into the product. So these are the two models of the enzyme activity. Now, enzymes functions, we know that enzymes have variety of functions, right? They carry out photosynthesis, they carry out signal transduction. So photosynthesis and cellular respiration, these two functions are important roles. The enzyme has an important role in the photosynthesis and the respiration. In addition to that, it helps in digestion. Signal transduction is nothing but cell to cell communication. Active transport, then various biochemical reactions. Inside our body, there are so many chemical and biochemical reactions goes on, whether it is digestion or it is maintaining homeostasis or the cellular respiration. Any activity, they require a lot of enzymes, and enzymes, they help in progress of chemical reactions, right? Energy production, ATP synthesis. So there are so many roles that enzyme plays in our body. So this just show you that how enzymes, they are important at various levels of digestion to convert the food, the digest the food and convert it into simple nutrients, right? So there are certain example like amylase is present in the mouth. Then you have the digestive juices like released by the pancreas, released by uh, the stomach. So you have different like gastric juices. So they have pepsin, trypsin. So these are all lipase. These are all enzymes which play an important role in digestion of starch, proteins, lipids, etc. Digested food, the nutrients are absorbed in the intestine, and the undigested food is released outside from the body. Now, what are the various factors which play an important role in enzyme activity? So, how do they increase efficiency or they will decrease its efficiency? So, temperature, pH, and substrate concentration are the important factor. So, now as we know that with the increasing temperature, what happens? With the increasing temperature, what will happen as the kinetic energy increases, the molecules activity increases. As the result of that, this will increase, the enzyme activity will increase, but it will, the rate of reaction will increase, but it will increase till this point, which is the optimum temperature. Optimum temperature is the temperature where the enzyme activity is maximum, right? What happens after that? As you increase the temperature, like suppose the 40-degree is the optimum temperature for this any particular enzyme. If you increase the temperature beyond that, what will happen? We know that enzymes are proteins, and we know that proteins, they get denatured when you raise the temperature, because what will happen, the with increasing temperature, the bonds will break down, like different bonds are present when the polypeptide chain folds to form the proteins. So these bonds will break down. Because of that, the active site's shape will change and it won't be able to react with the substrate anymore. So there is decline in enzyme activity. So the rate of reaction will also be decreased. Same pattern will be observed in case of pH. With the increasing pH, till the optimum pH, the activity will increase. But beyond the optimum temperature, if you go beyond, suppose the 8 pH is the optimum pH for the particular enzyme which has been shown here. If you go beyond that, the rate of reaction will decline. Now, the next concept is very important, like substrate concentration. That applies to substrate concentration as well as the enzyme concentration as well. When you are increasing one out of two, like when you are increasing, like you have the same number of enzymes, but you are increasing the substrate concentration. So earlier, what will happen? You have enzyme, more the substrate, higher will be the rate. So more product formation. But unless it becomes completely saturated, right? So what will happen? Earlier, it will increase, the rate will increase. So rate of reaction will increase. You have got this phase where now you have the, like here the reaction is now becoming saturated. So you have the same number of enzymes, but you have more substrate now. And when you have higher substrates, in that case, what will happen? Even if you have an excess amount of substrate, but your enzymes are limited. So now the reaction becomes steady. That becomes stable. There will be no more increase in the rate of reaction. Enzyme inhibitors are those substances which inhibit or interfere with the enzyme activity. There are two types of inhibitions, competitive and non-competitive. As its name indicate, competitive means it will mimic the shape of the substrate, and instead of, like, it doesn't allow the substrate to bind there, but your inhibitor will come and bind to this place before your substrate comes. So now your active site is blocked, so there is no space for the substrate to combine. As a result of that, no reaction, so it won't be able to further increase the rate of reaction because substrate is not going to bind to the enzyme, so no reaction. On the other hand, you have non-competitive inhibitor where you will observe that this is non-competitive, means it is not going to compete with the substrate, but it binds to a site which is a different site. That is what you call it as allosteric site. It binds to an allosteric site. Because of that, here the active site's shape will be changed. So there is a change in active site. As a result, it won't be able to bind. Further, you will find no reaction. So these are the enzyme inhibitors, and we'll quickly go over. This is how the energy, matter, and organization happens, that where the enzymes play an important role in the plant cell, in the animal cell, and they carry out the conversion of carbohydrate and water in the presence of sunlight into glucose and oxygen. And inside the human cell, or any animal cell, the mitochondria has a lot of enzymes. The glycolysis, Krebs cycle, and electron transport chain takes place. It produces ATP, and these nutrients will be recycled. So this is how the matter, energy, and this system is taken care of. Thank you.